Hello everyone, my name is Lucas Maia. I am a PhD student at Purdue University, and today I am presenting the results of the first year of a long-term study where I'm investigating the influence of cover crops on soil microbial activity and degradation of atrazine and S-metolachlor. The interest in the use of cover crops has increased substantially in the past years. Data from the state of Indiana shows that the number of hectares planted to cover crops increased 459% between 2011 and 2019, although this number represents only 3.2% of the agricultural area of the state. And nationally, the adoption of cover crops increased 49% between the ag census of 2012 and 2017, with cover crops being planted in a little more than 6.2 million hectares. Most of the cover crop use has been focused on the soil health aspect. The benefits of cover crops include increasing organic matter, increasing soil microbe activity, among several others. However, cover crops are also an essential tool for the integrated weed management approach. The physical barrier created by the residue, the competition for light, water, and nutrients, as well as the release of allele chemicals, are just some of the benefits of this practice from a weed control perspective, especially if we think about the challenging scenario with the herbicide-resistant weeds. Among the practices involved within cover crop management, termination is the one that results in the greatest impact on weed suppression. From the termination strategies listed below, herbicide application is by far the most adopted. A standard practice on termination of cover crops is the application of a non-selective herbicide, such as glyphosate or glufosinate, sometimes in combination with an auxin herbicide. And what has been proposed by several weed scientists is the inclusion of a soil residual herbicide at termination. And the idea is to provide some additional early season weed control and hence prevent weeds from competing with the cash crop. Like mentioned before, one of the benefits of cover crop use is the potential increase in the soil microbe activity. The soil microbial community is composed of fungi, viruses, and mainly bacteria. These organisms are responsible, for example, for decomposing organic matter, supplying nutrients to plants, and biodegradation of pesticides. In most cases, the microorganisms produce and release enzymes that will promote various chemical transformations in the soil. These are called extracellular enzymes, and one example is beta-glucosidase, probably the most commonly soil enzyme studied since it is responsible for regulating the carbon cycle in the soil. Another class of enzymes are the intracellular enzymes, and one example is dehydrogenase. This enzyme is present inside the cells of all living organisms within the soil, and therefore, in the presence of pesticides, these are the enzymes most likely to be directly affected. However, the response of a soil enzyme to a pesticide is unpredictable, since there are many other factors, especially environmental factors, that can influence the activity of these organisms. As well as cover crops, soil residual herbicides are included within the integrated weed management approach. These herbicides are most active in the soil, although some foliar activity can occur in certain active ingredients. Incorporation is a key factor on the weed control efficacy and can determine the success or failure of this class of herbicides. One of the key benefits of using soil residual herbicides is the early season weed control, which helps on reducing the selection pressure for herbicide resistance. And the degradation of these class of herbicides occurs primarily as microbial degradation, but chemical degradation is also common. Atrazine is the most common soil residual herbicide applied, and within all the herbicides, it is behind only to glyphosate in amount applied annually. Atrazine has a moderate absorption capacity to soils and has a half-life of approximately 60 days. Biodegradation is the dominant degradation pathway of this herbicide. However, in soils with low pHs around 5.5, chemical degradation becomes predominant. In addition to the natural biodegradation, enhanced degradation of atrazine was already documented in several states, mainly because of the repeated use of this herbicide. S-metolachlor is also widely used in corn and soybean production to control annual grasses and small-seeded broadleaves, and is the third most used herbicide in the US. S-metolachlor has an average soil half-life between 30 to 50 days, and its degradation is promoted almost entirely by soil microorganisms. Herbicide interception becomes an issue when applying soil residual herbicides onto cover crops at termination. Given the ground coverage provided by the plants, only a small portion of the herbicide applied goes straight to the soil. And in addition to the reduced amount of herbicide reaching the soil, the question that remains is that if cover crops are promoting an environment that increases the degradation of atrazine and S-metolachlor. The knowledge about biodegradation of these herbicides in cover crop systems is still limited and requires further investigation. Therefore, the objective of this study was to evaluate the influence of cover crops on soil microbe activity and degradation of atrazine and S-metolachlor. We hypothesized that the use of cover crops would increase soil microbe activity and atrazine and S-metolachlor degradation. To test this hypothesis, we conducted field trials at Throckmorton and Penny Purdue Agriculture Centers, which I will refer to as TPAC and PPAC. The experimental design of the field trials was a split plot design with 4 replications. The cover crops planted were cereal rye and crimson clover, and the fallow control was included. 3 levels of soil residual herbicides were tested within each cover crop and fallow, being no residual, medium residual, and heavy residual. All herbicides were applied following the recommended label rates and in-tank mix at cover crop termination. Soil chemical and physical properties were determined in the spring of 2020. The cover crops were terminated 3 weeks before corn planting, although we aimed to terminate them at 2 weeks, but due to rain we had to postpone the corn planting for almost a week. And the quartine rate and fertilizer applications were similar for both locations. Cover crop biomass was assessed the day before termination by collecting plants from a quarter meter squared. Soil samples were taken at 6 sampling times, starting the day before termination and at 3, 4, 8, 12, and 16 weeks after termination. These samples were then analyzed for beta-glucosidase and dehydrogenase activities, and also herbicide concentration. Herbicide concentration was determined following the Quatcher's method in a single quadruple LCMS, and the data from each site were analyzed separately and subjected to ANOVA using the ProcMix procedure and SAS 9.4. The means were separated using Fisher's protected LESD with Tukey's adjustment at an alpha of 0.05. Analyzing the results, we can observe that the use of cover crops did result in increased beta-glucosidase activity relative to the fallow treatment. In both sites, and in 3 out of 6 soil sampling events, at least one of the cover crop species studied resulted in greater beta-glucosidase activity if compared to the no cover crop control. In addition to that, we also noticed a clear difference in beta-glucosidase activity between the 2 sites. TPAC had an average of 2-fold more beta-glucosidase activity than PPAC, and that can be explained by 2 reasons. First, the cover crop biomass from TPAC was greater than that from PPAC, almost 2-fold in difference. Second, the organic matter content from TPAC is approximately 1.8-fold greater than that from PPAC. If we compare our results with some of the previous research, we don't see as much variation in the activity of beta-glucosidase throughout the growing season. In our case, that activity was relatively constant and seemed to be related to the soil temperature, which also did not show too much variation. As we can see in the dehydrogenase activity, this enzyme is not influenced by soil organic matter content and cover crop biomass the same way beta-glucosidase is. In fact, we could not detect a clear pattern in the dehydrogenase activity throughout the growing season. The use of cover crop did result in occasional increased dehydrogenase activity relative to the fallow treatment. And at TPAC, only crimson clover resulted in greater dehydrogenase activity relative to the fallow treatment at 12 weeks after termination. Before we analyze the herbicide concentration data, it is important to mention that this data was averaged across medium and heavy residual treatments because the rates of atrazine and S-metolachlor were similar between these 2 treatments. We can observe up to 88% reduction in atrazine concentration as a result of crimson clover use as cover crop at PPAC from 3 to 8 weeks after termination. Similarly, atrazine concentration at TPAC was reduced by more than 99% in cereal rye and crimson clover plots between 3 and 8 weeks after termination. Atrazine concentrations from samples taken between 12 and 16 weeks after termination fell below the limit of detection of the LCMS. The concentration of S-metolachlor in soil samples from the cover crop treatments was reduced by an average of 87% at T-pack and P-pack from 3 to 8 weeks after termination. We hope to better understand the impact of increased microbe activity and herbicide degradation as we collect more data during the next couple of years. Now, if we compare both herbicides, we notice that at 3 weeks after termination, the concentration of atrazine in the plots with cover crops was higher than in the fallow plots. For S-metolachlor is the opposite. Higher S-metolachlor concentrations were observed in the fallow plots relative to the cover crop plots. This may indicate that the use of cover crops may slow down atrazine degradation and enhance S-metolachlor degradation. To conclude, this research shows that the use of cover crops occasionally increases beta-glucosidase and dehydrogenase activities, but does not decrease relative to fallow. Also, increased soil microbe activity as a result of cover crop use results in enhanced degradation of S-metolachlor. The practical implications of this study are that the influence of cover crops on soil residual herbicide degradation varies among active ingredients, and the extended soil activity from atrazine might be possible when using cover crops, but more data is needed to support this conclusion. Data collection on this project will continue for the next 3 years at least. In future experiments, we will evaluate the fate of soil residual herbicides when applied at cover crop termination. With that, I would like to thank the support that has been given to this project by the Indiana Corn Marketing Council and also to all members of the Purdue Wheat Science team. Thank you very much for your attention. I wish you a great virtual meeting, and I'll see you next year, hopefully in person. Thank you.